Hi everyone, and thank you for the invitation to be here celebrating Dolph and speciation as well. So I'm very happy to be here, and today I want to share with you some of the ways in which we address some of the similar problems that you've been hearing so far. And I'm going to focus on plants and actually draw your attention as to whether you think similar things are happening or not across these type of major divisions in the tree of life. So I just wanted to start with this because I just wanted to highlight we're all here because we love diversity, and of course I wanted to put some sticklebacks there. I hope I got the right one, Dolph. And anyway, but you can see this diversity all across the board, and that observation is what intrigues us all. And that observation basically makes us ask whether natural selection has a role in the origin of organic variation and the discontinuity that we see, right? So this type of question is not easy to answer. It requires a lot of long arguments and many lines of evidence to be able to address it. And I'm gonna split the talk in two parts. The first one is like, how do we know actually that natural selection contributes to the origin of diversity and discontinuity, right? What is the evidence that we need for that? And the second one is, how does natural selection and inheritance work together to create this diversity and organic discontinuity? So let's get started with this one. And what I want you to think about it just like from first principles, our observation sometimes might tell us that things are obvious, right? I mean, like this particular tiger here, you immediately would think of camouflage. But that's not enough. I mean, that's an N of 1, there's a tiger there. And ideally what you want is to do experiments to demonstrate that your perception of what might be going on is actually correct. Now, on the tiger, perhaps it was a little bit obvious, but there are cases in which it stops being obvious, right? And this is the system that I work with. It's called Senecio lotus species complex. The Senecio genus is the most speciose in plants, and this is just one of those complexes. And I love it because taxonomically it's a nightmare. Nobody knows what to call what, and I think that's what we need to study, because as Dolph was saying, like it's just like divergence in action. Now, you can see here some very dramatic differences, like this dune type and headland type. They occur by the coast. But then if you go here to the right side, you have these two types that occur in the alpine regions of Australia, right? And those growth habits are very contrasting relative to the other ones, of course, like this tableland, for example, that lives next to the edges of subtropical rainforests, right? And now if you focus on the leaves, you have these extreme leaves, like filaments, versus these very elongated and serrated and lobulated. So the diversity is quite striking. And all of this is in glasshouse conditions. So we have removed environmental effects. And if you bring seeds from the field and you don't know where they were taken from and you grow them, you will realize which one of these ecotypes you're talking about. It's incredibly strong, the signal, okay? Now, Kirsten mentioned the idea that natural selection is a deterministic process. And I think that's key to be able to develop first principle predictions about what we should see about in nature. And one of the first things that we need to do is that to consider what happens as this determinism takes place, right? As this particular type of lizard is colonizing, the dark lizard is colonizing a different area where it's camouflage, etc. It might replace that particular one, and there's a switch in the genetic components, allelic frequencies change, phenotype changes, and of course, it is natural selection because it's correlated with fitness variation, right? So that'll happen probabilistically. Natural selection is just a probabilistic outcome of these three components, and it's deterministic. We can predict that black form will increase in frequency in particular environments. So the first prediction is that we should find a correlation between environment and trait. And that's a powerful prediction, but it could be that you just have an N of 1, right? You tend to find one particular, Okay, there he is. So you might just find this black lizard in this dark area and this white lizard in this white area, but it's just the first one that you found. And like the tiger, you cannot know. So the second layer of evidence is that because natural selection is deterministic, should you repeat the process over and over and over again, well, therefore, you should end up with similar outcomes as that process repeats, as it has happened in the sticklebacks and in the other systems that we've heard today. Now, you might have those two lines of evidence, but then the third layer is whether the strength of selection also matters with respect to the differentiation at the morphological level and reproductive level. So here we have two types that are two habitats, but not very different, and here two types that are very, very contrasting. So even if it has happened in parallel, right? Therefore, you still need to account for that strength of selection. And that would be another layer of evidence that would tell you about the deterministic processes, right? Now, experimentally, to use all of these lines of evidence, ideally you want to decouple the signal that comes from one trait from the signal that comes from other traits, right? It's difficult to do it with the wild populations and the natural populations because all of those traits are associated with one another, right? And I think that Dolph in his book has a chapter at the end in which there is a little paragraph that was about basically these deconstruction, reconstruction experiments. And actually, I fell in love with that idea, and I said, okay, we definitely have to do these things. And what we do is that we take the two ecotypes and we literally blend them with certain specific breeding designs in which we truly shuffle their genomes. And they are just like little particulate bits of each one of the two genomes. And then we can expose them to either artificial or selection in the wild to see whether that field site, whatever conditions are there, can reconstruct what we had before. And this is, of course, from a standard genetic variation, but it happens to be important, so that's actually quite good. So now, there are consequences of this as well, and this is perhaps one of the key issues here, and is that we have consequences of adaptation on speciation, right? And we have heard many arguments today that they should be connected, and of course, ecological speciation has been at the core and forefront of our thinking in these terms. So when we have systems of parallel evolution, and we have one ecotype here that has multiple independent origins of that particular form, and then the second one, which could be a derived one, like the lake types in the sticklebacks. Well, I mean, they might be compatible across ecologically and reproductively, right? Also these ones here, but it's very difficult to go between them, right? That is also a deterministic outcome of natural selection. So that's what we've been trying to do all across, trying to get and test these lines of evidence, these hypotheses about the deterministic nature of natural selection in this system. And we have been focusing on this coastal system, and I'll show you a little bit about it in a second. The alpine system, which also has these two growth forms, the tableland and the woodland types, because they are incredibly contrasting habitats as well. So these are the coastal ecotypes. On your left, there is the headland type. Is what we call, has a growth habit that is prostrate. The main stem basically never goes up. It just like lies on the ground. This is rocky environments, very salty. The soil bed is very, very tiny, right? So it's a very harsh environment. And the dune ecotype, that's the headland there. That's the little mountain that you can see there. And that's the dune ecotype that occurs in these sand dunes behind the beach. And they're actually tall. They have different branching morphology. They don't like salt. They don't like drought. They have a root that just goes down into the ground and extracts the water from the deep soil. Very, very different. And in the glasshouse, they retain these shapes. Now, what about, therefore, the first prediction, like correlation between environment and morphology? So here we have sample populations of dune and headland along the coast, and some dune populations here. There's only one headland population here on an island, but we cannot collect it. It's a protected area. And the first thing you can see here is that in multivariate space, if you measure many traits and you ask, okay, regardless from where you collect them, along the coast, what happens to the dune population, what happens to the headland populations? And they form these two big clouds. And when you look at one trait at a time, let's say vegetative height, all the headlands here on the right, all the dunes here on the left, they are consistently different from one another. So as you keep looking at that, there are many traits that show a strong correlation between habitat and the morphological variation. So that suggests that we might have a system in which perhaps natural selection has played a role. However, as we take a look at these population pairs, so for example, here you have a dune type and the headland type. This is another headland type that is actually south of Sydney, and this is maybe this one is around 700 kilometers north of this population. These ones are in between Sydney and that other population that I mentioned. This one is in South Australia. Like, this population is maybe 3,500 kilometers away from this one here at the top. And you can see here like the growth habits are consistently prostrate and erect. Now you have some like dramatic, for example, succulents in some of these, and that's a very highly plastic trait that disappears under glasshouse conditions. So the next line of evidence is like, well, can we actually say that beyond this correlation that we find, we see some repeated and independent evolution of these forms? And here we have a phylogenetic tree in which we have the dune populations and the headland populations intermingled, and this is consistent regardless of the markers that you use and the samples that you use, and it's an internal control that we have in the lab. Everybody that does an experiment in the lab has to build a phylogeny, and then just to see what they keep seeing, and it just keeps popping up. Like the snails, like the populations in one part of the coast versus another part of the coast are highly isolated from one another. And when we studied actually patterns of gene flow, we just didn't see gene flow neither between the pair, within the pair, or between the pairs. And it's something that I was highly surprised, but it makes sense in retrospect given the strength of selection that we see. In the past, there could have been some gene flow. These are populations that during glaciations, the coastline has moved, right? So that little mountain that you saw, the headland, sometimes could have been like hundreds of meters away from the new border of the ocean, right? So that has been happening all along. And therefore, we believe that in the past, there are some signals of gene flow, but no, it's not recent at all. Okay, so we have the correlation between environment and trait, and now we not only have that correlation, but we might have independent replicates of that. So now it's a fantastic tool to understand natural selection. And our transplant experiments, they also demonstrate that across the years, 12, 13, 14, 15, the direction of selection is consistent, but not the strength, right? So there's fluctuating strengths of selection over the years, but it's always the dune that's better in the dune, the headland that's better in the headland. Now, to get a feeling for whether the doing better has to do anything with the type of environment in which they occur, what we do is actually take advantage of this high diversity that we see at the morphological level and also the leaf type level. And we create these magic populations in which we basically force the variance in fitness to be 0 across the generations. So basically, every 2 parents contributes to offspring, and it's random, right? So we enforce and start with many founders, with 30 founders or more, and then we carry that variation over generations, right? So we're shuffling the genomes, we're deconstructing the ecotypes, and trying to harbor and maintain as much diversity as possible. So this is just to show how local adaptation works in these other ecotypes. And that's in the dune environment. This is the dune type, survives beautifully. The other ones effectively die. Then in the headland, which is a harsh environment for all of them, they die, but there's a small fraction of the individuals that survive. On the table environment, it's incredible. Like this tableland absolutely loves it, but the other ecotypes basically do very, very, very bad. And in woodland environments, which are ephemeral environments, these woodland populations only blossom and bloom when there is a good deal of rain. in fact, they don't do as well as even the tablelands, but it's the only ones that flower. The other ones just don't flower, and they seem to be completely mismatched with respect to the developmental channels that they are trying to do. They actually allocate their energy to producing leaves as if they were a woodland. But that doesn't work if you have the genes of a headland. Okay, now, if natural selection is driving differentiation here, what we expect is that fitness, which is here on the y-axis, right, should decline with how different the environments are. So here in the dune environment, here on the x-axis, there's a very different environment to the dune, and here 0, that is a local dune population where the transplant experiment took place. And these are 3 other dune populations, and then there are the tablelands, etc. This is the tableland environment, and as you can see, like the more different the soil it is, the less well you do by being that hybrid or that parental, right? So that suggests that there might be some particular environmental pressures, in this case have to do with the soil, that might be driving partially some of these negative correlations between environment and fitness. Now, we know that this is genetic because if we control for the proportion of local genes by doing like an F1 and the 2 back crosses, as Dolph just showed earlier, what we know is that the probability of establishment in the dune or the headland is proportional to the proportion of local genes. So it also seems to be an additive, kind of like genetic architecture for fitness. Many loci might contribute here. So overall, I mean, we have a case in which the divergence between the habitats really matters for the type of adaptation that we see. It's consistent across the years. And it seems that occurs not only within these focal ecotypes that we study, but also the ecotypes that occur in other areas, right? And we know now that one of the major elements in the soil that creates a very strong pressure is the amount of salt and also the humidity in the environment. Those 2 things, so drought and salt, seem to be very important. Now, we've been discussing, and I think Dolph illustrated beautifully this morning, like, I mean, but what happens to reproductive isolation? Sure, we have here local adaptation is very strong, but do you actually evolve reproductive isolation? So we're going back here to the idea of ecological speciation. You can cross within the ecotypes, but you cannot cross between the ecotypes, right? Reproductive compatibility within, reproductive incompatibility between. So here we have, again, some of the populations that we study. This is with another suite of markers. And you can see here some dune headland pairs that are in this part of the coast, and then these other dune headland pairs that are in Tasmania and also in this southern area. So what we did is we did crosses within this clade, and we corrected by the distance, phylogenetic distance, when we did crosses between the 2 clades, right? So we ask, okay, what happens within ecotype and between ecotype, within clade and between clade? So this is directly inspired by some of the work that Dolph has done in the past, and this is what we see. I'm gonna run you through this quickly, and here we have one form of reproductive isolation that has to do with fertility, and this one has to do with survivorship, with viability, okay? And the bottom, you have different clade or the same clade, and different ecotype, same ecotype, okay? So let's start with within the same clade. Now, if you are at this line, you are exactly as fecund or survive as well as your parents. Okay? And if you are above this line, you are reproductively isolated. And if you are below that line, you have some form of heterosis or hybrid vigor. So within a clade, yeah, look at these dunes and headlands. Same ecotype, dune, or headland. Most of them produce more seed than any other cross that you do within a dune or within a headland, right? Now, within that clade, between ecotypes, dune with headland or the headland with dune, you start getting a variance in reproductive isolation, right? You still have some hybrid vigor, some that are fully compatible, and some that start to be reproductively isolated. But the striking thing comes when you look different clades. And perhaps this here is the most obvious one. Different ecotypes, crosses, they're pretty much fully isolated. You cannot cross them. It's incredibly hard to get seed out of them. Just a little bit, some here and there, but super strong. And in here, which is dune with dune and dune with headland, if you forget about the colors, you get this spread, this variance in reproductive isolation, but they're actually structured. And if you have a mom that is a headland, yeah, you produce offspring that is way less the number of seeds than if you are a dune mom. In fact, the dune moms, when you cross them between clades, they produce more seed that they would produce within their own same population, right? So this is consistent with parallel ecological speciation, but this one is not. So the same ecotypes that are super convergent, they look very similar, they cannot be crossed. And I didn't show the data, but it's difficult to transplant them as well, the headlands, across headlands. So it seems that natural selection is definitely working, making them diverge. But within the headlands, natural selection, most likely mutation order speciation, in which uniform selection is randomly sampling similar alleles to find similar solutions in different ways, and most likely compensatory evolution, as it actually kind of try to fix the trait that is evolves, is what might create a lot of those incompatibilities. But different from what Dolph mentioned earlier on, this is not morphological. This is actually the standard sterility and inviability, right? Now, this happens also for inviability, yeah? And this trend from hybrid vigor to what the MAM does suggests that we have this scenario in which you cannot cross between, in this case, the clades. You need some time, basically, to be able to see it. The dunes, you can cross them despite 500,000 years of divergence, and they even are heterotic. But the headlands between clades are very difficult to cross. Very, very, very difficult to cross. So this doesn't negate the idea that natural selection is driving the evolution of these ones, but it's very different when you consider between ecotypes versus within ecotypes. It's just two different layers. Okay, so now we're going to try to say, well, but what about the genetics of this? What is the genetics of adaptation? And for that, well, I mean, finding the gene for a polygenic trait is tough. It's incredibly tough. So we have followed a very different approach. And in 2012, my student Federico Roda found that when we did rat sequencing and look at all of these pairs, like this is Coffs Harbour, Lennox Head, and so on, the different populations, you can find a subset of genes that have frequencies that are put all the dunes on one side and all the headlands on the other side, and it's a small fraction of the genome. But for the most part, the populations are just different in different ways, right? So we were really frustrated because I'm like, okay, well, I mean, this idea of one gene, one phenotype, definitely in 2012 was still kind of like, oh, it might happen, but ever since, I'm not convinced that it's going to be that easy, particularly for these polygenic traits. So one of the things that we notice is that even though the different genes are being recruited in different population pairs, they seem to share something in common. And it was that they have to do with predicted functions, of course, about reproduction of predicted functions, about the use of hormones in plants, okay? So what we found is that there were many genes that one way or another were controlling the biosynthesis, conjugation, transport, etc., of a hormone that is called auxin. And auxin is a super hormone. It basically does everything. And plant biologists sometimes get really frustrated when the answer is auxin, because you say, well, I mean, but it does everything. How are you going to know? But in speciation is awesome, because if it does everything, it can create links between traits. If it does everything, it can create developmental problems early on that might be solved in a different way later on in time. So anyway, we're looking at these ones, and these biological functions are overrepresented in 8 pairs, right? Nothing was in the 9 pairs, but some of them in 8 pairs, right? But then if you look at the nucleotide side, it's just like it just decays very quickly, the number of pairs in which they share. And at the gene level also, there are very few genes. But it's that set of genes that really matter. So in the sense that we had that hypothesis of different solutions to the same problem. But how do we get at it? We cannot get the genes. So what we did is the following. The premise was simple. OK, how did they discover those genes in the first place? And they did it with mutant screens in Arabidopsis, right? And they used these Petri dishes, a little bit of agar, put a seed, and the individual grows this big, and then they phenotype that, and then say, OK, I found the gene, and it interacts with B and with C, and they create a network. Now, what we did ask is, well, if I might not know what the gene is, but I can know that if some traits that are effectively the mutant traits that they found in the screens exist in nature, they would reflect divergence in that pathway. So what we did was effectively look at the mutant screens, at the different phenotypes, and then we say, well, I mean, can we find that phenotype in nature, in the natural populations? And there are phenotypes that, well, I mean, we had never actually thought of measuring because it's just such an alien kind of thing for us, working with the big plants and the big pots, etc., as opposed to just working under these conditions. But we gave it a go, and a developmental biologist colleague told me, look, I mean, your plant is tall and short, most likely has to do with the way the plant sends gravity. Right? I said, OK, that sounds fantastic. So we went and basically took populations doing headlands, and we said, well, we're going to do an experiment in which we're going to see whether the plant bends or doesn't bend in response to being rotated 90 degrees. Right? And what happens is that auxin controls tropisms in general. And the way it does it is, if I am the stem of the plant, I have auxins all around me. But if I get turned, then you create a gradient, and all the auxin goes to one side, and then more cells divide or elongate. And therefore, because this one is not doing anything, this becomes a knot, and then the other one hasn't necessarily bent. So that's the way it works. And this is the experimental setup. And we ask similar questions about this trait that, as we did before. That's an example of the setup in the CT room, in the controlled temperature rooms, and that is a gravitropic response, and that is an agraviotropic response. Okay. So remember, gravitropism, sure, but that's not the point. The point is that that difference reflects divergence in a pathway. I don't know which gene it is, but at least I know that is that construct. So here we have some population pairs. This is the difference in height between the dune and headland within the glass house and in the field. And here at the top, we have the gravitropic differences. And there's a huge clade effect. So when we look here at height, and here we have a gravitropism, tall plants have a stronger gravitropic response. But if you don't account for clade, you don't see anything. You see just literally like a blob of dots. But once you account for the clade, those two clades that I showed you before, then we have two types of responses that have evolved independently for the gravitropic response. So we have some, again, correlation between the trait and the habitat. And we have a parallel evolution of gravitropism as well, okay? But what we need to do is demonstrate that it's an adaptive trait. So what we did is we used this magic population. We crossed the population for 8 generations, and then you have these shuffled genomes. And then what we did is that we put them in the sand dunes environment, in the headland environment. Here you can see the little seedling growing. And this is painstaking because you need to literally glue on a toothpick a 3mm seed on the side so you can actually track the individual over time. And because we didn't know how fast they were going to die, it could have been in a week. So, well, at least it's by the coast. But we had to measure the plants in the morning and in the afternoon. The morning and the afternoon for a month, just to get the first indication of how strong selection was going to be. Then after that, we said, OK, every 2 weeks, and then we just every month, stabilized, etc. So what we then did is, well, I mean, we deconstructed the one in the middle, and that deconstruction is that magic population. And then we put it in the field, and we are not going to select by any trait. We're just going to let the fittest families go into the next generation. That's the only thing. We don't want to bias it for any trait, okay? And this is just one of the field sites where we do, and it takes 2 days to plant everything. Often it's in the like thousands of seeds, because germination rate also is tough. So we need to basically plant thousands and thousands of seeds. So it's really hard work, but in a beautiful place. And of course, I mean, like the family comes as well, and yes, fantastic. All right, so let's see what happens. So I'm going to tell you what we did. So we said, if natural selection has reconstructed some of these traits, and in this case, the gravitropic response that we found was evolving in parallel, then we would expect that the families that live the best in the field would produce offspring that looks like the local population individuals, okay? And the families that did really bad would have the alternative phenotype, okay? So here on the x-axis, I have that, the fitness of those families in the wild. And then we cross them in a glass house and we look at their offspring, and their offspring is in the y-axis, okay? So it's a covariance between fitness and trait, right? Like a little bit like Price's theorem, trying to apply that idea. Okay, so in the dune, so here at the top is gravitropic plants that bend, these ones don't bend. And this is what we see. In the dune, the plants the families that did very bad, they don't bend as much, and the families that did really well, they bend a lot. So we have segregated the variance for fitness. And because these are plants, we can keep the seeds. So it doesn't matter that they die in the field. I have a sibling stored in a fridge in the lab. So with quantitative genetics, we can account for that. And then we know that the ones that died, I can still grow them in the glass house. And that's what makes it so powerful. In the headlands, the strength of selection is incredibly strong. We lost the variability, basically. And basically, we got like a low, regardless of the fitness. But it's really difficult, basically, to work with hybrids that look like headlands. It's very tough in general. OK, so it seems that we have an adaptive trait. So if we have that adaptive trait, then we can understand the genetics of adaptation, because we just need to focus on that trait. If we need to look at the connection between adaptation and speciation, then we can focus on that adaptive trait and its consequences on reproduction. So just briefly, we have found something quite similar to what Dolph mentioned earlier, is that when we have this particular gene set that we have, it's called ABA3 and ODL, right? If they are homozygous, both of them, they don't respond to gravity. But any other combination, they just shoot up, okay? So there's something about being prostrate, being gravitropic. It's a very specific combination and it's homozygote, right? At least for these ones. And these genes, I mean, they act in membranes of the vacuole that actually imbibes water during germination, and as the embryo grows and bends, that water comes in, elongates the cell, and then therefore they can bend. So cell elongation per se might be a trait that is driving this. But also, auxin controls many things that have to do with reproduction, and I want you to remember this for something that comes afterwards. This is the way the magic population looks like. So each bar is the probability of assignment according to the genome of each one of them. This is the probability of assignment for the parent's dune, for the parent's headland. And you can see that we have maintained things at very intermediate frequencies. This is the GWAS that we did for the tails of the distribution for graviotropism. And there are some 39 kind of like hits that we have there. And this is a zoom of this area in linkage group number 9. And the interesting thing is that this particular hit here, which is the strongest hit and is next to this one, it's in a region of very strong differentiation. FST and DXY, it's just like, but it's like a block. So you know where I'm going now, right? So, and the diversity in that block is really low when you consider the headland populations, but is average when you consider the dune populations. And the headland is the derived one. Now, there's a huge block of linkage disequilibrium there in that region. And when we looked at, this is just 3D mapping of the structure of the genome, in which you can actually look at whether one piece of chromosome is like bending here and attached to another one, and they consistently, chromosome 1 and 7, talk to one another, right? But also it speaks of whether the DNA is coiled, supercoiled, euchromatic, heterochromatic. And we have found basically that same region here in the chromosome is actually a chromosomal inversion. But we also found in the other chromosome, not a chromosomal inversion, but a region of reduced recombination that is humongous, but it has nothing to do with an inversion. It's just like a super coiled bit, and it's not the centromere. Okay? So that inversion and the data that we haven't gotten on the GWAS, we have coupled it with the expression profiling of those individuals as well. And this is just a kind of a built model of what we think. We don't know which gene is driving the whole thing, but all of these genes are differentially expressed. All of them have to do with the regulation of auxin in some way. And we are basically now trying to build models borrowed basically from system biology, in which we can actually, sorry about this, we just got this last week. And each one of these genes has an interaction, and there's a differential equation that basically links the working of one node versus the other node. And if you start with here, then you end up with the last bit here, and that's bending of the plant, right? And that allows us to find sensitive places of the network. And by that, it's another way of mapping, basically, because we cannot map genetically. It's just like we won't be able to have the sample sizes for that. So we're trying to do other ways in such a way that we can demonstrate that there are sensitivities associated with certain genes versus others. Okay? So overall, the idea is that we have a hormonal pathway that we captured the divergence at very early stages in the embryo. And now you're stuck with that divergence, right? So in a hybrid, that same pathway is going to be recruited for branching, for flowering, for pollen production, and so on and so forth. So you need to navigate the pleiotropic effects of the divergence throughout the entire development. And at that point, well, you need to either compensate, as Dolph was saying, right? Or be different. You might actually look really, really, really different, right? So that, I mean, I just put this here because it reminds me of cellular automata. You have a little rule, and you repeat it and repeat it and repeat it and repeat it, and you end up with these wonderful patterns. Okay, so to wrap it up, remember that I show you that the auxin pathway also controls some aspects of reproduction. And what happens is that these aspects of reproduction are quite important because it led us to think that in this gravitropic distribution that we have in this magic population, what will happen if we do crosses with those that are agravitropic in this tail, if we do crosses within the gravitropic, and then between them. So as you show you within clay, between clay, it's the same idea, but with these traits, right? And within the headland type, you don't have failure. Within the dune types, like the gravitropic ones, you have a little bit of failure. So there's some segregation there of something. But between, you lose over 20% of the seeds. This is not like quantitative. This is like no seeds, right? If we do a quantitative measurement, it's even stronger, right? Like quantitatively speaking. So it seems that there's a genetic covariance. We don't know the genes again, but a genetic covariance between sterility and the gravitropic trait, which is adaptive. So as an incidental byproduct of adaptation to the environment, intrinsic reproductive isolation has evolved. Now, I think that the hormones actually are involved in this, right? And when a pollen grain arrives here to the stigma, to be able to grow through, a gradient of pH needs to be formed, right? And who creates that gradient is auxin dependent. And so what we did is we look at the difference in pH between the pollen donor and the stigma receptor and the seed set. And you can see there that the more different you are in the mom that produces you All right? There's something about the mum always. It produces this pollen, and that pollen brings something with it. It lands on an stigma, and if it's similar to the previous mum's pH, it grows. Otherwise, it doesn't. And the headland, here in green, always is the one that has the strongest problems. They are the pickiest, basically. And we think that it has to do with this ability to penetrate the stigma. Right? So pH differences seem to control the strength of the pollen receptivity and therefore possibly modulate reproductive isolation, right? And what we see is that this geographically also makes sense because when we look at pollen competition experiments in which we actually put either one single pollen type or the two pollen types at the same the same plant, and this is just a massive breeding design that we did and driven by Henry Arenas Castro, what we see is that parapatric populations tend to produce less seed set than allopatric, suggesting that there's some selective force driving this. But the most important thing is that the parapatric headland population, not the allopatric headland, but the parapatric headland populations are the ones that produce the least conspecific the least proportion of seed in the presence of two pollen grains. There's a cost for that female. Basically, they only can use the pollen from the same population. So all in all, we believe that natural selection drove the evolution of traits that confer adaptation to these local conditions, incidentally created, of course, extrinsic, but also intrinsic reproductive isolation. And it seems that not only drove that evolution of postzygotic isolation, right, but also is affecting multiple aspects of the development of the plant, as I just showed before, reproduction, but also the local conditions. And whether it's going to be pleiotropy or linkage, that's where we are at the moment. Okay, so that's our genotype to phenotype map and fitness map. We have these molecular traits that where we can actually study. They give rise to morphological and physiological traits that create that variation in fitness. And depending on these pathways, they're going to be recruited throughout development, and they are going to lead to differences in the individuals. Of course, this is not possible without a great lab and a great family, and this is just to say thank you to all of them. See, if it's going to take a little while, but hopefully it worked. It's a very big slide, I don't know. And and I wanted to just to say thank you to Dolph as well. I mean, I tried to do an AI-generated Dolph, but it didn't work. So that's my version of of Dolph, like who we're going to be studying in the future, I'm sure. So thank you very much, and I'm happy to answer any questions.